Hello everyone, I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is CRISPR-Cas9 Screens to Investigate Mechanisms of Cancer Drug Resistance, and our sponsor is Selecta. Our panelists today are Dr. Guotai Xu, a postdoctoral research associate working in the laboratory of Jose Baselga and Maurizio Scaltriti at Memorial Sloan Kettering Cancer Center, and Dr. Paul Diehl, Chief Operating Officer at Selecta. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar screen. And if you look to the tray at the bottom of your window, you'll see a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Dr. Xu. Please go ahead.
Thank you, Christy, for your kind introduction. Dear everyone, thank you so much for giving me the opportunity to share my work with you today. It's the most frequently occurring cancer in women, as shown in the areas in pink. In 2018, there were about 2.1 million newly diagnosed breast cancer patients and about 0.6 million deaths. Therefore, it is important to study and understand breast cancer. Throughout my PhD and postdoc work, I have been consistently interested in characterizing the genomic drivers that drive breast cancer drug resistance. Breast cancer can be divided into several groups based on the expression of estrogen and progesterone receptors, and whether they have amplified HER2. ER-positive tumors have the expression of ER. HER2-enriched tumors have amplification of HER2, and triple-negative breast cancers are negative for ER, PR, and HER2. ER-positive tumors are primarily treated with anti-ER endocrine therapy, either alone or in combination with CDK4/6 inhibitor or mTOR inhibitor. HER2 tumors are primarily treated with anti-HER2 therapies. Triple-negative breast cancer can be treated with immunotherapy, anti-TROP2 ADC, or PARP inhibitors. Here, I want to give you one example from my previous studies in which I performed CRISPR screens to identify genes that affect endocrine therapy response. The screen result is shown here. Guide RNA library transducing cells were treated with either DMSO or ER degrader fluorescein, and then genomic DNA of each group were subject to NGS to retrieve integrated guide RNAs. I plotted the log2 mass of reads of each guide RNA in the DMSO group against the log2 abundance of each guide RNA between fluorescein versus the DMSO group. Each dot represents an individual guide RNA. From this plot, we can clearly see that guide RNAs against the ARID1A were highly enriched in the fluorescein treatment group versus the DMSO treated group, suggesting that ARID1A loss may mediate fluorescein resistance in ER-positive tumor cells. This study has already been published, so I'm not going to give you more details about it. Today, I am going to talk about our unpublished data on mechanisms of resistance to PI3K alpha inhibitor. About 40% of ER-positive tumors have PIK3CA mutations, which can be treated with PI3K alpha inhibitors. This therapy option was approved based on the phase 3 study, in which a significant improvement of progression-free survival was observed in patients who received the combination compared to those who received only endocrine therapy fulvestrant. However, intrinsic or adaptive drug resistance is frequently seen in the clinic. So, studying mechanisms of resistance to PI3K alpha inhibitor is a research priority. We have previously shown that loss of PTEN causes resistance to the PI3K alpha inhibitor in the clinic, but it only partially explains the resistance mechanisms in patients. In addition, we uncovered that sustained activity of TOXI1 limits the sensitivity to PI3K alpha inhibitors. Our following studies, as well as other group studies, revealed that the activation of PDK1-SGK1 axis, IGF-1RP110 beta activation, or PIM1 activation, may serve as the molecular mechanism of sustained TOXI1 activation. Moreover, we have previously shown that enhanced ER function upon PI3K alpha inhibition may also limit the sensitivity to PI3K alpha inhibitors. And later, we proved that the enhanced ER function upon PI3K alpha inhibition is through KMT2D, which induces active chromatin state for ER binding. Lately, we uncovered that double PIK3CA mutations in cysts increases sensitivity to PI3K alpha inhibitors. In this project, while CRISPR Cas9 guide-RNA knockout screens, we aim to unbiasedly identify and elucidate potential genetic mechanisms of resistance to PI3K alpha inhibitors. Briefly, a 150K guide-RNA library targeting the human genome was transduced into Cas9 expressing MCF7 and T47D cells. Here, different colors represent different guide-RNAs. Then library transduced cells were expanded for 3 weeks to allow efficient genome editing. These cells were treated with either DMSO or PI3K alpha inhibitor for 3 weeks, and the genomic DNA of each group were subject to next generation sequencing to retrieve integrated guide-RNAs. Further, bioinformatics analysis allowed us to determine those dropped out guide-RNAs and enriched guide-RNAs in drug-treated cells compared with DMSO-treated cells. The results are shown here. I plotted log2 mass of risk of each guide-RNA in the DMSO group against log2 abundance of each guide-RNA between PI3K alpha inhibitor versus DMSO group. Each dot represents an individual guide-RNA. From this plot, with this criteria for top hit selection, first the top 2.5% enriched guide-RNA, and each gene should have at least 4 guide-RNAs were enriched. We can clearly see that guide-RNAs against the many negative regulators of TOXI1 were highly enriched in the PI3K alpha inhibitor treatment group versus DMSO treatment group, including subunits of TSC1 complex, GET1 complex, histone complex, and the potential components of MPK-mediated TOXI1 signaling, suggesting that convergent TOXI1 activation may mediate resistance to PI3K alpha inhibitor. Potential location of those candidates in the TOXI1 signal pathway is shown here. All of them are potential negative regulators of TOXI1, which integrates intracellular and extracellular signals and play a central role of cell metabolism, growth, proliferation, and survival. And here is a simplified cartoon of what we saw from the screens. TSC1, TSC2 complex acts as a GTP activating protein gap for the small GTPase rep, and it is a critical negative regulator of TOXI1 by stimulating the conversion of rep GTP to inactive form rep GDP. AKT1S1 negatively regulates mTOR activity in a manner that is dependent on its phosphorylation state and binding to 14-3 proteins. STK11 is a key upstream regulator of MPK by mediating phosphorylation and activation of MPK. MPK directly phosphorylates TSC2 and receptor. PD7A is one of the CMP phosphodiesterase family members, which hydrolyze CMP to produce AMP. The GET1 complex, together with the histone complex, inhibits the loading of the TOXI1 to lysine, and thereby inhibiting TOXI1 signaling. To validate the screen results, first we CRISPR knock out each candidate in MCF7 and T47D cells, as shown by Western blot. For the validation of those candidates, we employed cell competition assay because it is more sensitive and more efficient. Briefly, GFP-labeled wild-type cells were mixed with RFP-labeled wild-type cells or knockout cells at the ratio of 1:1 and treated with DMSO, alpelisib, or tacrolimusib for 6 days. The change of GFP-RP ratio was scored as the impact of candidate gene knockout on cell growth and drug response. Our data shows that knockout of most candidates does not affect cell proliferation, with the exception that TSC1 and TSC2 knockout in MSKCC cells and TSC2 knockout in T47D cells. Consistent with the screen data, we observed that loss of most candidates limiting the sensitivity to alpelisib in both MSKCC and T47D cells, with the exception that C12OF666 loss does not cause resistance to alpelisib in MSKCC cells, and the MARK2 loss does not cause resistance to alpelisib in T47D cells. I will tell you the reason later. Similar findings can also be seen with another PI3K-alpha inhibitor, tacrolimusib. Since each candidate may negatively regulate TOXI1 signaling, next, we wondered whether TOXI1 activation is sustained in knockout cells in the presence of alpelisib. Consistent with the role of each candidate in TOXI1 signaling, we observed that TOXI1 activation is sustained in most knockout cells in presence of alpelisib, with the exception that loss of keto complex does not significantly affect TOXI1 activation in MSKCC cells, consistent with the relatively small effect on PI3K-alpha inhibitor sensitivity. Similarly, we also observed that TOXI1 activation is sustained in most knockout cells in presence of alpelisib, with the exception that loss of TBC1D7, AKT1S1, PDE7A, or MARK2 does not significantly affect TOXI1 activation in T47D cells, consistent with the relatively small effect on PI3K-alpha inhibitor sensitivity. We wondered whether the sustained TOXI1 activation is independent of PI3K-AKT activation. Interestingly, according to the previous Western blot result, the AKT signaling is still efficiently inhibited, suggesting that the sustained TOXI1 activation is independent of PI3K-AKT activation. Moreover, we observed that knockout of those candidates confers resistance to AKT inhibitors, confirming that the sustained TOXI1 activation is independent of PI3K-AKT activation. But how about mTOR inhibitor? To this end, we used mTOR inhibitor everolimus, and we observed TOXI1 activation is efficiently inhibited in knockout cells and wild-type cells. Consistently, knockout cells show similar everolimus response to wild-type cells as determined by cell tight glow assay. Since the sustained TOXI1 activation confers resistance to PI3K-alpha inhibitor in knockout cells, next, we wondered whether mTOR inhibitor could reverse TOXI1-mediated PI3K-alpha inhibitor resistance. To address this, we employed crystal violet-based colony formation assay. Consistent with the previous competition assay, we observed that knockout of each candidate confers resistance to PI3K-alpha inhibitor or AKT inhibitor, but not mTOR inhibitor. Most importantly, co-administration with mTOR inhibitor could reverse the resistance to PI3K-alpha inhibitor, suggesting an alternative treatment strategy for tumors which developed the toxin-1-mediated PI3K alpha inhibitor resistance. So far, we have found that knockout of each candidate from the screen confers resistance to PI3K alpha inhibitor through maintaining toxin-1 signaling, which can be reversed by co-administration with mTOR inhibitor. To our knowledge, the correlation between the GET1 complex and the cancer drug resistance is largely unknown. So next, we wanted to further dissect the role of the amino acid sensing toxin-1 signaling pathway in mediating PI3K alpha inhibitor resistance. Briefly, activation of toxin-1 signaling pathway by amino acids is mediated in part by the RAG GDP. The GDP-GTP ratio bound the status of RAG heterodimers, consisting of RAG-A or RAG-B stably bind to RAG-C or RAG-D, determines the localization of toxin-1 complex and thereby its activity in the amino acid sensing pathway. RAG heterodimers have 4 possible nucleotide binding states, but are active for the toxin-1 complex binding only when RAG-A or RAG-B is GDP bound, and the RAG-C or RAG-D is GDP bound. The GET1 complex has been shown to stimulate GDP hydrolysis by RAG-A-B that switch RAG-A-B from the active form to inactive state. So, loss of GET1 complex upon DEP-D5, NPR2 or NPR3 knockout may increase levels of GDP-bound RAG-A or RAG-B and therefore lead to constitutive activation of toxin-1. Therefore, we next implored whether inhibition of nutrient-sensing toxin-1 signaling restores DEP-D5 knockout-induced PI3K alpha inhibitor resistance. To this end, we overexpressed dominant-negative RAG-A GDPs, RAG-A mutant, or dominant-negative heterodimer RAG-A, RAG-D in DEP-D5 deficient cells. Observe that overexpression of RAG-A mutant or RAG-A, RAG-D dominant-negative mutant in DEP-D5 depleted cells abolishes the toxin-1 activity as determined by Western blot and blocked DEP-D5 knockout-mediated drug resistance to PI3K alpha inhibitor and AKT inhibitors. Actually, overexpression of RAG-A mutant alone was sufficient to blunt the toxin-1 activation in DEP-D5 knockout cells, indicating that the deregulation of RAG-A or RAG-B GDPs induced by DEP-D5 knockout is responsible for those phenotypes. Our ongoing work is to explore the clinical relevance of our study. We have previously shown that sustained activity of toxin-1 limits the sensitivity to PI3K alpha inhibitors in PIK3CA mutated tumors. Molecular mechanisms that cause mTOR activity and drive resistance remain to be fully elucidated. There are limitations regarding the clinical validation. For example, there are a limited number of pre- and post so-called paired PIK3CA inhibitor-treated samples, and alpelisib was just approved by FDA last year. Second, there were only a few genes which were included in our MSK Impact NGS platform, including TS1, TS2, and STK11, but not others. Third, the genomic lesions of those candidates are rare in PIK3CA mutant ER-positive breast cancers. Across this large-scale breast cancer genomic data, we observed that mutations of each candidate in PIK3CA mutated ER-positive breast cancer patients, and those mutations might have, mutations are mutually exclusive with each other. We are analyzing a group of genes which handles clinical validation, and as we all know, the prior therapies for patients may matter. far, we have analyzed our patients who were involved in this alpelisib clinical trial. Interestingly, we got 2 patients who developed resistance to alpelisib and acquired TSC1 or STK11 mutation. Now we are checking whether such TSC1 or STK11 mutation is a loss of function mutation by putting each back in TSC1 or STK11 knockout cells for PLK3CA inhibitor sensitivity study. We performed the whole genome-wide CRISPR screens to identify negative regulators of the toxic-1 complex that affect the PI3K alpha inhibitor response. This sustained the toxic-1 activation in knockout cells upon PI3K inhibition is independent of PI3K AKT. Co-administration with the mTOR inhibitor everolimus reversed toxic-1 mediated PI3K alpha inhibitor resistance in PLK3CA mutated ER-positive breast cancer. Loss of GET1 is a novel mechanism of resistance to PI3K alpha or AKT inhibitors. And in the clinic, we also found 2 patients who acquired mutations of TSC1 or STK11 who developed the PI3K alpha inhibitor resistance. I would like to thank my postdoc supervisor, Dr. Maurizio Scatteretti, and all our group members for their contributions to my project. I would like to thank my great collaborators, especially Yan and Fan. I was a convergent scholar supported by SU2C Foundation and the V Foundation. Also, our work is supported by NSF, NIH, and the Cycle for Survival. Thank you so much for your attention, and I'm happy to take any questions.
Thank you, Dr. Xu. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'll conduct the Q&A portion of the webinar after the presentations have concluded. Next up is Dr. Deal. Please go ahead.
Thank you, Christy. And thank you, everyone, for tuning into the webinar today. And thank you, Guotai, for sharing your experience using CRISPR screening to investigate adaptive drug resistance in breast cancer. Before we take questions, I'd like to give a very brief overview of who Selecta is and what we do. We're a small technology-focused company just south of San Francisco in Mountain View, California. We're not a venture company. Our company started about 12 years ago with seed money from research grants raised by our founder, Alex Chenchek. We use these funds to develop technology for products and services for functional genetic screening, to help identify genes responsible for disease progression, drug resistance, cell differentiation, development, and other biological processes of interest. In particular, the goal was to create technology platforms that enable broadly parallel simultaneous analysis of large numbers of genes at once, genome-wide analysis. Our goal also is not to develop technology that requires large-scale instrumentation or LIMS systems or other industrial infrastructure that's only available to a few large labs, mostly pharma and biotech. Our aim is to provide tools that can be used by a wide range of more traditional laboratories, basic research labs that are familiar with molecular and cellular biology. We've been able to do this by leveraging the rapid development of next-generation technology, next-generation sequencing technology over the past decade. As next-generation capabilities have increased, so has the ability to deconvolute results from complex biological assays and measure simultaneously thousands of assay elements in parallel. Using NGS technology, the readout makes it possible to pull out results from thousands of individual reactions and track changes in multiple samples, treatments, or time points. We've had some success with this approach. Here are some publications for over the years that have cited our technology from customers or groups that we've collaborated with. We have a more complete and updated list of citations on our website. The link is shown on the slide. I would encourage you to take a look at the site. It has a search function, and if there are any particular applications, cell systems, or procedures you're interested in, you may be able to find relevant studies that would provide some resources to set up your experiments. The first platform I'd like to talk about today is the driver map expression profiling assay. This platform enables you to get data on which genes are activated or deactivated in different samples from different treatments. cell culture conditions, biopsies, other types of patient samples, etc. Probably the most common approach to look at changes in gene expression on a global or genome-wide scale is RNA sequencing. While RNA sequencing is a very powerful approach, it is also somewhat technically complex with some challenges in prepping and running samples. And also, since it requires reverse transcribing and sequencing the whole transcriptome, data analysis requires somewhat intensive bioinformatics to deconvolute the sequencing results and determine the expression levels of each transcript. You really need to establish a robust pipeline involving several individuals and different specialties to effectively run RNA sequencing on large numbers of samples. For expression profiling, actually though, for small numbers of genes, the gold standard is still really qRT-PCR. It's very straightforward and quantitative, and it gives you robust, accurate measurements of each targeted transcript. With the driver map platform, we've tried to combine the simplicity and sensitivity of qRT-PCR, but do this on a more global genome-wide scale to give you comprehensive data similar to RNA-seq. We were able to successfully do this by developing a set of 38,000 primers, 19,000 PCR primer sets that amplify targeted segment of all 19,000 human protein coding genes in a single multiplex reaction. So this actually was quite a lot to develop. But as a result now, starting with total RNA, you can run a single RT-PCR reaction and get 19,000 amplicons, each unique for a different human gene. And then the pool of amplicons can be quantified on next-generation sequencing or by next-generation sequencing. As a result, the advantages are, first, it's very easy to run, especially as compared with RNA-seq. A single RT-PCR reaction with targeted primers so that only total RNA is required as input. You really don't need to do mRNA enrichment, beta-globin depletion, or other types of manipulation. You can start directly with total RNA. It's very sensitive since it is PCR-based. It provides robust genome-wide profiles for very small samples down to picogram amounts. We routinely run the assay on samples with single or just a few cells. While 19,000 amplicons may seem like a lot to sequence, it's much less complex than RNA sequencing, the RNA sequencing analysis, which requires analysis of the whole reverse transcribed fragmented transcriptome. As a result, we get much deeper sequencing and stronger reads of low-expressed genes with even 5 times fewer sequencing reads. We typically run, for example, 5 million reads per sample with the driver map assay, and for genome-wide sequencing with RNA-seq, you need to run 25 million reads or more in order to get similar types of depth. Finally, the analysis is more straightforward. Since the 19,000 amplicons are defined and can be directly aligned to the expected reference samples, the read levels for each amplification directly correlate to the expression level of the gene. As a result, the data from driver map assay can be easily analyzed on a spreadsheet. So, depending on what you're interested to understand in your system, driver map can provide a much more straightforward and effective approach to profile and correlate genome-wide changes in gene expression with different phenotypes of interest. While driver map and other expression profiling techniques provide an understanding of which genes are up and down regulated in conjunction with the changes or presence of different phenotypes, transcriptome analysis does not tell you the genes important for driving phenotypic changes. Which genes are activated or deactivated that cause the biological changes of interest? To address this question, it requires disrupting or perturbing particular genes to see if they have an effect on the phenotype of interest. There are really only 2 technologies that enable this sort of gene disruption analysis to be done on a whole genome-wide level in a single assay, RNAi and CRISPR. This is because with both of these technologies, it's possible to use a single common method and gene database to design and make sets of very similar reagents, shRNA hairpins or guide RNA for CRISPR, that can be introduced into a large population of cells and disrupt and or otherwise perturb different individual target genes in each cell. As Guotai spoke about, the CRISPR pooled screening approach is a very powerful approach to identify genes responsible for driving biological changes. The most common application of this approach is the one that Guotai discussed, using CRISPR sgRNA to identify genes that are essential for cell viability. With any CRISPR screen, the approach is basically the same. First, package the library as lentiviral particles and then transduce i.e. infect, your target cells with these lentiviral particles so that you have each of the sgRNA in multiple cells in the population, and mostly just a single sgRNA in each cell. If you are looking for an essential gene screen then, if you are looking for an essential gene, then the sgRNA that are targeting an essential gene in a cell will be toxic to that cell, and they will kill it. That cell will die and drop out of the population, so that after a period of time, these toxic sgRNA targeting essential genes will be underrepresented. It's a reverse or negative selection. The depletion is detected by isolating genomic DNA from all of the cells and then amplifying and sequencing the sgRNA guides by NGS. The guides that are underrepresented then are presumably targeting essential genes required for viability. As Guotai showed in his presentation, this sort of screen is useful to identify genetic vulnerabilities in cancer cell populations. Also, it's often used to identify genes that confer resistance to therapies. Other screening approaches, such as looking for overrepresented sgRNA in a population or using FACS to sort cells based on a reporter or cell marker, can also be utilized to look at genes required for activation of a particular pathway, or required for a drug's factor or mechanism of action. Can also be utilized to look at genes required for activation of a particular cell pathway or for a drug or factor's mechanism of action. The technology that we use to make the sgRNA and shRNA libraries is not limited, of course, to those types of cells. To those type of DNA sequences. Using some cloning tricks with defined pools of oligonucleotides, we can construct libraries that have millions of defined sequences, also known as barcodes. These can be transduced and used to uniquely label hundreds of thousands of cells in a population. Since barcodes are on lentiviral vectors, they are integrated into the genome and are heritable. So you can create a founder population that, as they propagate, all the progeny from each of the barcoded founder cells are also genetically labeled with barcodes. This type of labeling can be useful for a number of applications. For example, to look at cell heterogeneity and cell pools in a population, to see how the population of cells change over time with differentiation or implantation. Also, whether certain subpopulations in cells have specific characteristics that increase or decrease their ability to survive drug treatments or other selections. Actually, researchers from MD Anderson will be discussing this application of the barcoded libraries in our next webinar in September. Also, on a single cell level, with a single cell platform, such as the 10X system, genomic barcodes in the cell can be correlated with the activity of particular genes or changes in specific expression profiles and grouped by subpopulations or sets of founder cells. It's also possible to add sgRNA into the mix too and look at how different subpopulations respond or are created when certain genetic changes are purposely induced. This is related to the perturb-seq, crop-seq, or CRISPR-seq methods that are sometimes discussed if you're familiar with these approaches. And so, this was intended to be a quick overview of the capabilities Select offers. If you're interested in discussing any of these technologies further or getting more information about the products and services we have available that incorporate them, please feel free to contact me. Again, I would like to thank you for tuning into the webinar, and I hope you found it interesting, and I'll turn it back over then to Christy.
Okay, I believe we're now live for our Q&A. Thank you, Dr. Deal, for your presentation, and Dr. Xu for your presentation. So as a reminder to webinar participants, once again, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to take a moment after the webinar has ended to give us feedback by taking our exit survey. All right, so our first question, Dr. Xu, could you go over again how you did your gRNA screening and what your output, how you evaluated your output to select the candidate? Guotai, you might be muted. Yes.
Yes,
We go.
now it should be okay, right? Okay.
Yes.
So, yes. Can you see this slide?
Yes, we can.
Okay. Yes. So here is the scheme that I did the screen. So first we got the whole genome guidance library from Selecta that's targeting the human genome. In this library, they have about 8 guidance targeting each gene, so total about 160K. And then I package them into lentivirus using, this is the normal lentivirus packaging system. And then I use the MOI, which is less than 0.5, because we do not want to have so many integrations per cell. In Cas9 integrating MCF7 or P470 cells. So it's just a normal lentivirus infection procedure. And then we use puromycin to select the positive transduced cells to get the library transducing cells. And then this is very important because we need to wait at least 3 weeks. Based on my experience, and also it's cell line it might be cell line dependent. So we have to wait at least 3 weeks to have efficient genome editing. And then we get the cells with gene knockout, individual gene knockout. Then those cells can subject to either DMSO or PI3K alpha inhibitor treatment for 3 weeks. So this is also dependent on the drug or the cell lines. So we use 3 weeks drug treatment, and then you will have 2 groups. One is the DMSO treated group, and the other one is the PI3K alpha inhibitor treated group. Then you get the genomic DNA from each group for PCR amplification to amplify the barcode. Here the barcode means the guide RNA, because each guide RNA has different sequence. So you can just amplify the guide RNA, and for next generation 16, retrieve all the different guide RNAs, and you will get the reads of each guide RNA. And then you can do comparison by bioinformatics analysis too. So it's very easy. So we'll compare each guide RNA from the PI3K alpha inhibitor group versus the DMSO treated group. You will know which guide RNA was selectively enriched or dropped out. So then you can see the second slide. So you can see those guide RNAs, the top right part, all the dots, they are selectively enriched. And the bottom right, that means there are a lot of guide RNAs that were dropped out. But in this project, we only focus on the enriched guide RNAs, because for those guide RNAs, you will identify which genes whose loss causes resistance to PI3K alpha inhibitor. So basically, this is the scheme, and of course here I use this stringent criteria for top hit selection, but it's all dependent on your experience. So here I use this hit selection criteria. Yes.
Great. And can you tell us what the efficiency is of the inheritance of the CRISPR barcoded cells?
Efficient of what?
The inheritance of the CRISPR barcoded cells?
The barcode itself, what does that mean? So you mean the efficiency of knockout or efficiency of what? Sorry.
I believe the questioner is asking how efficiently are the CRISPR barcoded cells? How efficiently are they taken up?
I don't know whether you can say efficient of the barcode, because their DNA, so they are very, very stable. But if you are talking about the knockout efficiency, it really depends on different guide RNAs and different genes. Yeah. So for the barcode, it's very stable. I think the efficiency is because you will use the puromycin to select the positive transducer clones. So they're always there because this is a stable transduction. So it's infection. It's integrated to the genome. So it's always stable. Yeah.
Okay.
And they won't lose. Yeah.
All right.
Yeah, maybe just to add to that, they're in a lentiviral vector, so the lentivirus integrates into the genome, which is what Guotai is saying, and then it's stably in the cells, and so all the progeny from those cells also get that.
Okay, great, thank you. Paul, we have a question for you. Does the driver map profiling technique also amplify long non-coding RNAs?
No, the format of the assay that we have right now is targeted specifically to protein coding genes. That is one difference between the driver map approach and RNA sequencing, is that with RNA sequencing, you're looking at really the whole transcriptome. With driver map, you need to have, you're targeting specific sets of genes, and so we've targeted all protein coding genes. We could develop an assay for long non-coding RNA or other sets of genes, but at the moment, the assay is focused on the protein coding genome.
Great, thank you. Guotai, have acquired mutations of GATOR1 been observed in clinical samples that would be consistent with the results that this is not only an mTORC1 inhibitor that also may contribute to PI3K alpha resistance?
Yes, this is a very important question. So here in the paper, we try to, the novelty of this paper is we found the loss of GATOR1 causes resistance to PI3K alpha inhibitor, and as we all know, the GATOR1 was recently discovered by David Budin's group. But the function of GATOR1, the correlation of GATOR1 in drug resistance has not been fully elucidated. So this is very, very important. So the short answer is no. We do not see any acquired mutations of GATOR1 in those patients who acquired resistance to PI3K alpha inhibitors. I think one reason is that we do not have enough prepared clinical samples. And another reason is that the genomic mutations of GATOR1 are very rare in ER-positive PIK3CA mutated breast cancer patients. So, but now we set out to do immunohistochemistry to check the protein levels of GATOR1 subunits in patients who acquired resistance to PI3K alpha inhibitors. Yeah.
And then in slide 10, the CRISPR screen, that you mentioned in slide 10, you mentioned looking for enrichment or dropout of the guide RNAs, but it seems like you only found enrichment. Can you tell us why that is?
Slide 10. Oh, I think maybe you mentioned slide 15 because the first 5 are some introductions. So I think still this slide. Yes.
So your slide 10. Yes.
yes. Of course. When we make this slide, we always want to say from the same screen you can identify both enriched guide RNAs and also dropped out guide RNAs. So from those enriched guide RNAs, you will identify genes whose loss causes resistance to PI3K alpha inhibitor. However, from those dropped out guide RNAs, you will identify genes whose loss may cause synthetic lethality to PI3K alpha inhibitors. So both are very, very important. So in this mTOR paper, so today I will only focus on this paper, the mTOR paper. So we only focus on the enriched guide RNA. Actually, we have another one ongoing project focusing on the dropped out guide RNA. In this paper, only enriched. Yes.
I see.
I'm sorry, Paul, did you want to jump in as well?
No, sorry. No, okay.
All right.
Yes.
Guotai, would you say that alpelisib and alpelisib are just different versions of the same basic inhibitor, or is there a difference between them that's bigger than that?
Yeah, that's also a very, very good question. So in this paper or in this project, we took alpelisib and alpelisib as the same drug. However, they are not the same. They are not exactly the same. So as we all know, the class 1 PI3Ks consist of heterodimers of P110 catalytic subunits and also P85 or P101 regulatory subunits. So for the P110 catalytic subunits, they have four different isoforms: PIK3CA, PIK3CB, PIK3CG, PIK3CD, which include P110 alpha, P110 beta, P110 gamma, and P110 delta, respectively. So there are just 4 different isoforms. So for alpelisib, this is a very selective inhibitor of P110 alpha, so the alpha isoform. However, alpelisib is a potent inhibitor of alpha, delta, and also gamma. So not P110 beta, so called P110 beta sparing inhibitor. Yes, so they are kind of different, have different function, but they both target P110 alpha.
And on your slide 14, so I guess that would be our slide 19 in our lineup, when you did the targeted CRISPR knockouts of the genes in the mTOR pathway in the MCF7 and T47D cell lines, did you use synthetic RNP single guide RNAs or a viral construct? And then also, can you tell us, did you select clones or did you use cell pools?
Yes, that's also very, very important. This is the reason why I like the construct from Selecta. So, yes, we always use both RNP system and viral system for our experiment. For this Western blot result, this shows the knockout efficiency of each gene with the viral system, and we use this for our competition assay in the following slides. However, for example, on slide maybe 36, I can show you this one. 36, I think. Yes. So for this slide 36, we use RNP system for this experiment because we need to put the mutant, the reg mutant to the DPDC5 knockout cells. They both have pure myosin selection marker, so we cannot use the viral system to do this. Otherwise, you will have the same selection marker. So we use the RNP system to save the pure myosin selection marker, and then we use the pure myosin resistant reg mutant to those knockout cells to do this for this. So this is very, very important. And also, as I mentioned in the clinical validation relevance part, we also need to use the RNP system to knock out TSC1 or STK11, and then we are checking whether such TSC1 mutation or STK11 mutation is a loss of function mutation by putting them back in TSC1 or STK11 knockout cells for PIK3CA alpha inhibitor sensitivity assay. So both assays we always use. So this is very, very important. And for your second question is about whether we pick out the single cell colonies or we use the pool the population. The answer is we always use pooled population because we think the pooled population could faithfully represent the whole population. And as we all know, because of tumor heterogeneity, each colony cannot represent the whole population. So that's why we always use the pooled population. And with the system from Selecta, we think, I mean, at least in my hands, it's kind of easy to have very good knockout efficiency with the pooled population. I don't think it's a problem. But of course, it depends on the gene. I think maybe some genes, if the gene is very, very, for example, it's toxic, maybe you cannot get a very good knockout. But for the genes which I always work, they're kind of okay for the whole population to have good knockout efficiency. Thanks.
And are there any controls that you use to evaluate a guide RNA abundance before treatment?
Guide RNA abundance before treatment?
Mm-hmm.
I do not really understand this question. Guide RNA abundance before treatment? You mean the T0?
I think Guotai, I think it's referring to the screen and how the guide RNA is. What's the distribution of the guide RNA before you actually run the screen, or how is that determined?
So
how do you determine depletion or enrichment? I guess is the relative to each guide.
I guess maybe the question means the before treatment. I think maybe like in the middle part, the cells with gene knockout. So to see whether they are stable after DMSO, I mean, or after untreated. So just to compare to see whether I think maybe it's talk about the toxicity of each guide RNA, I guess. So yes. For the toxicity, it really depends on the gene. For example, normally I have the control PCNA, it's an essential gene, and the guide RNA targeting PCNA for sure will be dropped out. And actually, this is exactly the reason why I said I had to wait for at least 3 weeks for efficient genome editing. Actually, I used the control of PCNA and also other essential genes because we found 3 weeks later, the guidelines targeting PCNA or other other essential genes beta actin, they can be efficiently dropped out. So it's really dependent on gene. If the gene is toxic, then the guideline for sure will be dropped out automatically, even without drug treatment. I think this is probably the question they ask, I guess.
Right, and for the ones that you're interested in, then you're looking at the dropout in the treated population versus the untreated.
Yes.
So it doesn't really matter how represented that particular sgRNA is in the whole population overall. It's just you're comparing the number you get from
that's
untreated versus treated.
Yeah.
Okay.
That's not a problem. Yes, because you only compare the DMSO versus drug treated group. Yes, of course for some that's not a problem at all.
Okay.
All right. Thank you both. We don't have any other questions in the queue, so I guess we'll wrap it up there. We'd like to thank Guotai Xu, Paul Deal, and our sponsor, Selecta. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this genome webinar.